active transport. So understand that this is going to be part of molecular transport because you are transporting a certain number of molecules, and this does depend on the gradient. Okay. In fact, active transport is interesting because at least the primary active transport is responsible for creating the gradients. Okay. So it actually creates gradients, but we'll get to that in a minute. So again, you're going typically against the gradient, okay? And that means you're going from where a substance is low to where the substance is high. So active transport, the most important word about active transport is that it's a pump, okay? So it's a pumping mechanism. So I want you to really visualize this because the pump is really important. Okay? And active transport is mediated primarily by carriers. And the reason for this is that when you're trying to get something impossible achieved, right? Like pushing a material from low to high, you're going to need the most powerful machine out there. A channel is not going to cut it. Does that make sense? Channel type motivation is not going to cut it. Plus, remember that active transport requires energy. Now, in physiology, that is always ATP. Okay? So in a physiology class, that's always ATP. If you're in my biology class, I'd be singing a very different song. Okay? In fact, ATP is made by active transport that doesn't use ATP. Think about it, if you're making ATP, why would you use it? That doesn't make any sense, right? Yeah. So in mitochondria, everybody should know this from their biology classes. In mitochondria, remember the last step of electron transport chain when hydrogens come down their ATP synthase molecule, right? The coming down of hydrogen ions will help you spin the wheel that makes the ATP. If you don't know what the heck I'm talking about, don't worry about it. Okay? But there are examples where active transport can occur without ATP being used. In fact, active transport causes ATP to be made. Am I making sense? In the mitochondria. So if you don't know this, don't worry about it. You don't need it. But if you know this, please appreciate that active transport is not always ATP requiring. You can use other molecules to do this. So it requires energy. In fact, when we do the digestive system, we'll look at an example like that, where hydrogen ions are used to transport fatty acids. Fatty acids and, not fatty acids, I'm sorry, I take that back. Tripeptides, not fatty acids. Tripeptides in the intestine are transported using hydrogen ions, not ATP. It's active transport, but it is transported using hydrogen ions. Hydrogen ions are like a bomb. I mean, the intensity of energy in chemistry, the smaller something gets, the more powerful it gets. Is that clear? Okay, so we'll get to that then, but for now, just understand that I'm giving you the simple model, okay? It requires energy. Most of the time, it's in the form of ATP. And those of you who don't know this, remember that ATP is hydrolyzed to ADP and phosphate plus energy. And ADP is hydrolyzed to AMP and phosphate and energy. Also understand that ATP is a neurotransmitter. Again, it's easy for you to think that's all ATP does. That's all it does. Okay? No. Remember, all hail evolution. Every molecule has a million functions we don't know about. Are we clear? Yes. Always work with that humility or you're not going to learn jack. Okay? If you keep your mind closed and think this is all it does, okay? That's what fundamentalists do, is that's all they want to see, right? You're a biologist. You cannot think like that. Remember, this is why, an eczema medication can cause depression. Right? The whole purpose of your drug discussion is for you to appreciate that it's never a single pathway. Do you understand?
Yes.
Never, okay, never. You don't need a million years of evolution for that, okay? So this energy is really, really important, and this is what's used for most of the cellular functions. And ATP is the energy currency of the cell. When you stop producing ATP, the cell has been dead for at least half an hour, okay? The cell's already dead when you stop making ATP. All right, everybody okay with this? All right, so how does this work? They're carriers, right? Which means what must happen?
They must carry. Transfer.
Get more, that's the right answer. They must carry, yes. But to carry, what must happen? Shape change, conformation change, thank you. It's extremely important. You must go through shape change. Okay, so there are going to be several examples, okay, of active transport that we're going to go through. Several examples, okay? But before we go through that, I want to go through the two types of active transport. So primary active transport, like I said before, is constitutive, meaning it never turns off. It's always on. It turns off when you're dead, okay? Second of all, it creates gradients. I don't like the word creates. It establishes gradients. Okay, or sets up gradients, you can say that too. Establishes is more scientific. So establishes gradients, primary active transport, fuels secondary active transport. Okay, and it's usually an all-in-one pump. All-in-one, meaning there is a pocket, and the correct term is domain. So every protein can have many domains, just like every body can have different parts. Every protein, the parts of a protein is called a domain. You need to know that. Parts of a protein is called a domain. So each protein can have many, many domains. RNA polymerase, if I recall, has 26 to 27 domains, okay? Huge, okay? It's one of the biggest proteins we know. And some RNA polymerases, I don't know if it's human, but some of them, highest recorded, 256 domains, okay? Just amazing, all right? So all these parts, they have to work together, correct? So what are the parts of a primary active transport mechanism? You have a built-in ATPase. Anything ase is an enzyme. Anything ase is an, I'm going to go very quickly, so you have to ask me if something is unclear. You have a built-in ATPase, meaning it breaks down ATP and traps the energy very efficiently. So these enzymes are very efficient. You have a built-in ATPase. And I'm going to draw a cartoon for how this ATPase works. The example we're going to use is the most universal example, which is the sodium, potassium, ATPase pump. Okay? This is a primary active, this is how you describe something. Primary active antipoder. What does that mean? Good. You're transporting multiple substances in opposite directions and you're using energy, correct?
Yes.
So give me a complete definition. Okay, so there you go. That's important. So how does this work? Okay, so it's a cycle. So there's no end point, start point, right? You just arbitrarily start somewhere. So I'm going to start with the membrane. So here's the membrane, okay? And here is the ATPase pump. Everybody with me? Draw as I draw, okay? So here we go. So this is the pump. Now, this is the cytoplasm. Whenever you draw the membrane, if you do not put cytoplasm and ECF, that diagram is meaningless. Do you understand?
Yes.
Because which one are you talking about? Right? You have to orient the membrane, correct?
Yes.
You have to orient the membrane, correct?
Yes.
Mm-hmm. All right. I'm on my 9th hour of babbling. If you do not answer, I'm going to just sit and take rest. Okay? And if the syllabus goes behind, You're gonna have to learn that stuff on your own using my videos. I will test you on it. Is this making sense?
Yes.
So it's in your best interest to get all that energy, especially the Thursday class, at least the Tuesday, they suffered with me for 3 hours. Thursday, you're coming here from a nap. You need to answer. I know it's not a nap. I'm just making fun of you. I'm jealous that you didn't have to teach 9 hours. But you have to answer. Please participate. Okay, keeps me going. All right, so here's the pump. The purple is the pump. Everybody got that?
Yes.
All right, now first thing that happens is so first of all, understand that in a cell, okay, I need you to orient this is important. Your sodium ions are very low inside and they're very high outside. You see the size of my letters?
Yes.
Do you see that gradient? That's a gradient. It's a difference, right? Potassium ions are huge inside the cell and they're tiny outside the cell. Got it?
Yes.
And there are numbers. You don't need to know the numbers just yet. Okay. So keeping this in mind, 3 sodium ions will bind. Okay, 3 sodium ions will bind the sodium binding pocket. These are proteins. Do you expect them to be specific?
Yes.
Proteins are extremely specific, okay?
Yes.
So sodium will only bind that pocket. Are we clear?
Yes.
So you got 3 sodiums binding that pocket. What this will do is the sodiums are positively charged. Everybody agrees?
Yes.
ATP is a charge of -9 because it has 3 phosphates. That's hella negative. Are we clear?
Yes.
So ATP is immediately attracted to the sodium. But it takes a little time for it to get there, correct?
Yes.
So keep that in mind, important. So what happens is, and again, this is a very dumbed-down structure, right? Clearly, it's a very complicated looking sculpture. I'm not drawing that, right? So this is a dum-dum level, okay? So ATP will bind its pocket. ATP will not bind until the sodiums bind. Are we clear? This is called the induced fit model of enzymes. Otherwise, you'll be wasting ATPs, okay? Sodium binds, then the ATP binds, okay? So on this side, I'll be writing this description. Okay, so let me write what's going on here. So first, 3 sodium ions bind on the cytoplasmic side. 2, ATP binds. Okay, 3, ATP broken down by ATPase. It binds the ATPase. The ATP is broken up. In reality, what happens is when the sodiums bind, that's a 3+ charge, right?
Yes.
That's going to change the shape of the protein so that now the ATPase pocket opens. Till then, the ATPase pocket is closed. Can this bind ATP? No.
No.
Okay?
Yes.
Can I lift my glasses with this?
No.
No. This is the shape I want, right?
Yes.
So when sodiums bind, look at my hand. When sodiums bind, boom. If sodium doesn't bind, I can't lift it. Are you seeing this?
Yes.
You can learn this entire class using your hands. Okay? No, really.
Yes.
Can you believe 1 year Berkeley gave me the Outstanding Professor Award in 2003 just for hand use in my classes? It was hilarious. I thought it was a joke initially, so I didn't go to the function. So they actually gave me the award. And I thought it was because I had won it 2 years in a row already, so I figured they were just joking, so I didn't go.
They have to hunt you down to give it to you?
No, they mailed it to me. They're like, you didn't come, here. But use of hands is that useful. Use your hands, okay? So ATP broken down by ATPase. Number 4, okay? This energy, okay, inverts the pump. Very, very important. So this energy from the ATP breakdown will cause you to invert the pump. So again, here's the membrane, right? Now what is the pump going to look like? Yeah, wrong color.
Reverse.
Right? Pump is inverted. Everybody see that?
Yes.
But what else does it have? It has your 3 sodiums, right?
Yes.
Okay, the direction may be off. Yeah, the sodium should be on this side. Doesn't matter, I'm not wasting time. So ATP is broken down, right? ATP is not there, but instead of an ATP, all you have is a phosphate. Okay, the phosphate is there. as long as the phosphate is there, the pump is inverted. Are we clear?
Yes.
So let me ask you this. This is how my exam questions will be. If there is a mutation in the ATPase pump, okay, I don't see people writing this down. Exam question, hello. If there is a mutation in the ATPase pump where the phosphate cannot be removed, understand me? The phosphate cannot be removed. Okay? What will happen to the sodium and potassium levels inside the cell or outside the cell? What happens to those gradients?
The gradients will slowly wither away and eventually.
Yep. The gradients will cease to exist. Is that making sense? Because what am I saying in the most fanciest way? If that phosphate cannot be removed, is this pump useful anymore?
No.
The pump is poisoned. Are we clear?
Yes.
It doesn't work anymore. Okay? The gradients will cease to exist and life will cease to exist. Are we clear?
Yes.
So those kinds of mutations will never be supported through evolution. Okay? So there's your phosphate. So what's the next step? So do you see what I'm doing? I'm writing the stuff on the side while I'm drawing. Okay? I would do that. It's very, very useful. Okay?
Yes.
So 5, the phosphate is removed. And who does that? There are, again, enzymes, phosphatases that are built in. Okay? This is really interesting. These phosphatases, you don't need to know this, but the phosphatases are activated by the potassiums. The potassium from the outside will activate the phosphatase and potassium binding. So first of all, the sodiums will leave. Okay? So what have you effectively done? You have transported sodium out, correct?
Yes.
Okay? So then number 6 as a step is, so I'm going to say number 5 as a step, potassium, 2 potassium ions bind. True or false? Potassiums bind the same site as sodium.
False.
False.
I would say false. I actually don't know for sure. Okay? I haven't researched this. My guess would be it's false because the protein is big enough to have designated sites, and sodium and potassium are different sizes. Are we clear?
Yes.
Very different sizes. So I doubt if they'll bind the same spot. So 2 potassiums bind, right? And then what ends up happening is once the phosphate is removed, the pump flips back. I'm really simplifying this for you. It is never this simple, okay?
Yes.
It's a huge dance, okay? I'm not choreographing that, right? So we don't have time, so I'm just telling you that in essentiality, it flips, okay? So you get the protein to become like this, right? So potassiums bind, right?
Yes.
2 potassiums bind, and so the 2 potassiums, okay? Shouldn't draw it there, should draw it here. The 2 potassiums are easily drawn into the cell because the proteins in the cytoplasm are negatively charged. They will attract the potassium. Potassium is positively charged. Are we clear?
Yes.
So what attracts sodium technically outside are the proteins that are around the cell. Remember that there are proteins in the extracellular fluid and not a lot, but sodium is powerful enough to be attracted to them, right? Potassium needs the strong attraction of proteins in the cytoplasm. So it all works out, okay?
Yeah.
So then number 6 is in our description. Number 6, inversion, okay, of the pump. Okay, 7, 2 potassiums dissociate. I'll give you a minute. Please look at this and tell me if there's anything there I can explain. Did you get my notes from last week on Canvas?
Yes.
Okay, good. If you're not on Canvas and checking every day what's going on, this class is going to be tough for you, okay? Even if you've had physiology, because you have no idea what I'm putting there and what you need to know. Okay, so make sure, checking Canvas all the time. No questions?
No.
So is this simultaneous or it's like,
You tell me. 1 through 7. Can this be simultaneous?
well, I'm thinking now.
The ions have to come, bind that slot, right? It all takes time. It cannot be simultaneous. Which means primary active transport is always slow. They're very, very slow. Is this making sense?
Yes.
This has tremendous consequences, especially in relaxation of skeletal muscle. If this pump was not, or this type of transport is not slow, your muscles would snap and break your bones. All hail evolution. If anybody sees this, that's what I'm going to say. All hail evolution. Is this making sense? All hail evolution. You know, my class made me a mug saying all hail evolution. So the bottom line is the functioning of the pump is very, very slow. I've said this 4 times. You're not writing this down. The less you write down, the less I write down. Okay? We're going to see the effect of this most likely today. If not today, definitely on Thursday. You're going to use that when we do action potentials.
Most active transports are slow. Most of these functions are slow.
All primary active transporters are slow. They have to be slow because you have to do all this dancing. So it's not just important to understand they're slow. What's more important? Understand?
Yes.
I mean, yes. Understand what?
The distance about
It's not enough to understand that something is slow. It's even more important to understand, finish the sentence,
Why it's slow.
why they're slow. Okay, good. You have to understand why they're slow. Okay, so very slow mechanism. All right, primary active transport done. Any questions on this? The sodium potassium ATPase pump is the reason sodium ions are low inside the cell and high outside the cell. So when we started out, sometime during our embryo, sodium was equal on both sides, right? But believe me, the egg, right after its fertilization, the first thing that starts working is sodium potassium ATPase. Everybody thank your mother, because that's where you get that one from. Okay. Moving on to secondary active transport. Secondary active transport is very, very specialized. Very specialized. Are we okay with that? I'm going to start with an analogy, then we go to the actual transport. Okay, so bear with me. So secondary active transport, okay, this is very interesting. It is called an electrogenic pump. Electrogenic pump. It does not use ATP directly. ATP is what kind of energy? Is it electricity? Is it mechanical energy? Is it chemical energy? What is it? ATP. ATP is electric. What voltage are you getting out of ATP? It's chemical because it's bonds. 102 days, 102 days. ATP is chemical energy. So primary active transporters use chemical energy. Secondary active transporters use electrical energy. Very different deal. Is that making sense?
Yes.
Very different. Okay? So secondary active transports cannot be uniporters. I will ask this on the exam, guaranteed. If you do not see that question on your exam, you're taking an economics exam, get out. Okay? If this is not on my test, okay, either I should be admitted to some facility, or because I've lost it, or you're taking the wrong exam. Secondary can never be uniporters. Okay, so let's talk about why. Okay, let's see how this works. Let me give you the analogy first, but before I give you the analogy, let me explain the principle of how this works. and most likely you won't understand it at all. That's fine. Then I'll explain it. So the principle of how this works is cations are transported down their gradient. Is this active transport? No. Okay, they're transported down their gradient. This releases electrical energy. That's the definition of electricity, is movement of positive ions down the voltage gradient. That is the definition of electricity in physics. Okay? So it releases electrical energy. Take it from an AP physics teacher. So releases electrical energy. So you release electrical energy. That's why it's called electrogenic pump. Genic means giving birth. Hello?
Yes.
Okay. So this electrical energy is used to pump another substance. Hint, another substance. Can this be uniport? No. That's why it's never a uniport. Okay? Used to pump another substance against its gradient. And we're going to see why we don't like saltless chips. All hail evolution. Okay? Let's see. So, when, against its gradient, not gradients, okay, gradient, that's only one. So, what is the analogy? The analogy is this. Let's say I have a gasoline driven car. If I don't do anything, the car just sits there, correct? Or you should go push it, right? Nobody wants a hernia, so no one's going to push the car. Forget it, right? So when will the car move? When I burn the gasoline in my car, correct? And you initiate that with an ignition, but ignition alone won't move your car, right? The gas has to burn. If you ignite and there's no gas in the tank, the car's not going to move. Am I making sense to everyone?
Yes.
Not a tough example, right? So what are we doing there? The energy you get from burning the gasoline is used by the car to push the car forward or backward or however you're driving. Am I making sense?
Yes.
That's exactly what's happening here. Okay, so I'm going to draw it. So what is happening here is this. So here you have a cell. Secondary active transport is not ubiquitous, meaning it's not found everywhere. Primary active transport is ubiquitous, particularly sodium potassium pump, is ubiquitous. It's found on every living cell. So how does secondary active transport work? Now, if you look at what's going on here, so let's say that, for example, this is an intestinal cell, right? This is a cell. This is one of the cuboidal cells, or you can say it's the columnar cell in the intestine, but I need some space here, so I'm going to draw it as a cuboidal cell. So this is the hollow of the intestine called the lumen. You need to know that word. And this is the basolateral side. And basolateral is where the blood vessels are. B for basolateral, B for blood vessels.
And what's the lumen?
Lumen is the hollow of the intestine.
Okay.
So if you take a tube, okay, this is the lumen. Everybody got this?
Yes.
Okay, and this is the basolateral side. Lumen, basolateral side. Everybody got that?
Yes.
Use your hands. Okay. So the secondary active transport. Let's talk about primary active transport. I told you, if a cell is alive, and in the kidney you find some exceptions. But if a cell is alive, it will have the sodium potassium pump. Only the kidney shows an exception. There are some cells that don't have the sodium potassium ATPase. Instead, they have potassium hydrogen ATPase, right? For a very different reason. Are we clear? So we'll get to that when we get there. That's just an exception. Everywhere else, you're going to find sodium potassium pump, right? Everybody, all hail sodium potassium pump.
All hail evolution.
So let's draw that transporter. So here it is. So I'm going to draw it, keeping my color code. I'm going to draw it in purple. Yes? Everybody see the purple ball?
Yes.
What does that do? It pumps sodium out, correct? And it pumps potassium in, correct? And this is happening all the time, which means sodium is going to be inside the cell. What? Sodium is going to be blank inside the cell.
Out.
Low. Sodium is going to be very low inside the cell. Good. So sodium, oops, wrong color. Okay, sodium, half my life goes playing with color on notability. So sodium is low inside the cell, correct? Everybody got that?
Yes.
Okay. Now, the secondary active transport, okay, I need a different color for this. Let's see, I'll pick this orange. Okay, so let's say you have, this is the secondary active transporter. It is super stupid to have primary and secondary active transport right next to each other. Do you understand?
Yes.
Yes.
That doesn't work, okay? And in the kidney, you will learn why, okay? But this happens there. We're talking about the intestine, same story. You don't want them on the same side. So what this pump does, keeping the color code, sodium is high in my food, correct?
Yes.
Why is sodium high in my food? Don't say it because you're Indian, although it's true. Why is sodium high in my food? I'm gonna retire in a minute.
Salt.
Salt!
Yeah.
Do you eat saltless food? Who
No.
eats saltless food?
No.
Even in deserts, you put salt. Show me one cookie that doesn't have sodium. Because if you made cookies without sodium, it doesn't taste good. You always add a pinch of salt into your cookie dough. Hello?
Yes.
Yes.
Oh, cookie dough. I'm becoming a Homer Simpson now. Cookie dough. All right, so sodium is always, even if you don't have sodium in your food, your blood has plenty of sodium. Sodium is always around. Am I making sense? If sodium is not there, you're dead. End of story.
Yes.
So sodium is high outside. Everybody got this?
Yes.
Okay. Now, so what would sodium want to do? Go inside the cell, correct?
Yes.
Can it go inside the cell on its own?
No.
No, you need a bridge, right?
Yes.
And the only bridge here happens to be this orange looking channel, correct?
Yes.
It's not a channel, it's a secondary pump, right? So sodium will go in carefully. And when sodium goes in, it's a positively charged ion. It's going from an area that's positively charged to an area that's negatively charged. So you're going down your voltage gradient. When a positive ion goes down a voltage gradient, blank is generated. Electrical energy or electricity is generated, correct?
Yes.
So what do you use that electricity for? And I'm going to use another color. There you go. Life being wasted on colors. I'll use this light blue, okay? So glucose is low inside the cell. Can you see that? Sometimes that monitor is awful.
Glucose.
Yeah, you can kind of see it. Sorry. Can you see it?
Yes.
Okay. So glucose is huge inside the cell. Can you tell me why? You eat,
Yes.
and insulin is working, correct?
Yes.
Yes.
What is the purpose of insulin? I screamed about this all, my vocal cords will cry if you don't answer this. What does insulin do? Take blood sugar into your cells, correct?
Yes.
So glucose is always going to be high inside. If it's not, then your insulin is not working. Homework, I want you to work on this and tell me what happens to this in diabetics. Because there, glucose is low inside the cell. It's not that easy. Go home and think about it, okay? It is not easy. That's your homework. Go home and think about it, okay? I'm not going to talk about it here, because I ain't got the time. Okay, here we go. So what happens to glucose? Glucose gets pushed. Okay? I'm going to make it thick to show you that it is pushed. Are we clear?
Yes.
You get the energy from the sodium coming down, and you use that energy to push the glucose, correct?
Yes.
In Niagara Falls, okay, or in any of the dams. Okay, there's one in Las Vegas. I forgot the name of that thing, because I always think about casinos. There's a dam there, right? Hoover Dam, yeah. So what happens there? Water comes down from a certain height and turns on the turbine, right?
Yes.
And that turbine generates your electricity for you, correct?
Yes.
The same idea. You're generating electricity by making sodium come down, and that energy is trapped. You need somebody to do all this, right? That's your secondary pump. Are we clear?
Yes.
that will push glucose. So here's my dance, right? So sodium is coming on my left side. Everybody say yes?
Yes.
Okay, sodium is coming. This will give me energy to push glucose, push glucose, push glucose. Do you see this?
Yes.
You do this 3 times, you will never forget this concept. And on the exam, you can do this. Okay? All right, so next thing you know, I'll fall without the chair. And then I'll go home. So, do you see how the secondary pump works?
Yes.
You're not using ATP directly. Is ATP involved indirectly?
Yeah.
Yes, because if this pump were to not work, this sodium will never be low.
No.
If this sodium is not low, this won't happen.
Yes.
If this don't happen, this don't happen.
Mm.
So if your sodium potassium pump is unable to function,
Like in glucose.
okay, secondary will not function. Is this clear?
Yes.
Okay? So how do you write this in bougie language? This is how the bloody Queen of England will write it. Okay? So here it comes. So glucose, and the same with amino acids, same with fructose, same with any secondary active transport. So glucose is pumped into cells, and when I say pumped, that means against or along.
I guess.
Against, okay, pumped. It's pumped into cells using electrical energy generated by facilitated diffusion of sodium ions into cells, made possible by maintenance of low sodium concentrations, and square bracket means concentrations, intracellularly by the sodium potassium ATPase primary active transport pump. Tell me if that doesn't summarize the whole damn thing.
It does.
That's what you learn in 8 years of grad school. This is what my questions will look like. So the reason I'm writing all this is to familiarize you with my language. Are we clear?
Yes.
This is how I think. So I'll give you a minute to write down. Is this concept making sense though?
Yes.
Okay. I'm just going to mention this. You don't need to know this just yet, but you will need to know this eventually. So in muscle, all types of muscle, no exceptions, skeletal, cardiac, smooth, doesn't matter. In all muscle types, we have a calcium ATPase unipolar. You need to know this, okay? So, which one? Okay, calcium ions are pumped out by calcium ATPase uniporters. Is this a primary or secondary mechanism?
Primary.
It's a uniport. Cannot be secondary, remember?
Yes.
So this is going to be a uniport primary mechanism. Everybody got that?
Yes.
So calcium levels are 0 in the cytoplasm. You need to know this. Calcium levels are 0 in the cytoplasm. You do not want any calcium there because the minute calcium sees phosphate, it's going to precipitate. And you're going to actually do these experiments in lab next week, I think. Okay? So calcium will immediately precipitate phosphate. You do not want that in the middle of your cell. Are we clear? Because if you take phosphate out, can you make ATP?
No.
No. Okay? Instant death. Okay? So we don't want that. So cells go through a lot of effort to keep calcium out. And calcium is a big ion. It's not going to enter very easily. So tell me, would calcium leak channels ever exist? That's disaster. Okay? Calcium leak channels don't exist. Are we clear? There's sodium, there's potassium. You do not want calcium leaking. Okay? Are we clear?
Yes.
Too big of an ion. So molecular transport is over. Okay? We're moving into bulk transport. This one's easy. It's basically you just need to know what the different mechanisms are. Okay? And I have already listed what bulk transport is. So you have the definition. I'm going to now list out further classification. Okay? So bulk transport, so finish this chart. Okay? So bulk transport, I'll do it here. Okay? Because I don't have enough space there. Bulk transport is very, very important to physiology. It happens all over the place. You're transporting big, bulky substances like a bacteria or a protein, big things, okay? Or you're transporting one huge mountain. That's a bulk transport, right? It's only one mountain, but mountain will not fit in your car. Am I making sense? You have to have a truck. Or you're transporting 2 billion little pebbles. You're probably going to need a truck for that too, correct?
Yes.
Even though they're small pebbles, right?
Yes.
So in bulk versus bulky, you need to know this. So in bulk as well as bulky, you should know the difference between the two. Water molecules, would that be classified as in bulk or bulky?
In bulk.
In bulk, water is a tiny molecule, correct?
Yes.
So what does bulk transport involve? There are 5 requirements. Okay, this is a vesicular transport. Okay, so you need vesicles, and vesicles have proteins. Did this thing die? Yeah. They have a rechargeable one inside. Okay, but no. We don't have, they will not give us access because I'm going to run out with all of that and sell everything on Pine. So pissed. I have to use this ghetto piece of junk. There's a nice one inside. Okay, that fits right in my mouth. So cool. Watch, this will last me exactly half an hour. So fed up with this. Okay, I think that's how it goes. Doesn't even say which way it goes. There. Yay! You can hear me now, right? This thing may screech. Okay, well, I turned the volume up just a little bit. All right, vesicular transport. So vesicles have specific destinations, okay? They need to go to specific parts of the cell. You learn all this in bio. So vesicles, okay? Vesicles absolutely require, vesicles walk on the cytoskeleton. You know what cytoskeleton is, right? Set of fibers inside the cell. Vesicles literally walk on them, okay? So for vesicular transport to happen, what do you require? If you're using a freight train to transport something in bulk, you're going to need train tracks, correct?
Yeah.
Okay, those train tracks are your cytoskeleton. So you need your cytoskeleton to be active. And cytoskeleton is only activated by calcium. So you need calcium for this. So requirement number 1, vesicles. Requirement number 2, cytoskeleton. Requirement number 3, calcium. Okay? Now tell me, does the Costco truck care about how many pasta packets you have in your garage?
No.
No. Bulk transport doesn't give a damn about your gradients. Are we clear?
Yes.
Gradients are not important. So gradients are irrelevant. Okay, now do you need energy for a Costco truck?
No. Yes.
Yeah, I mean, you could walk to the Safeway or Costco to get your pasta, but for the Costco truck, no chance. You're gonna need energy, right? Either a battery operated truck or gasoline, right? So energy is a must. So people who are not ardent cell biologists will classify bulk transport as active transport. In fact, your useless book actually says that. And I'm going, what blasphemy, okay? Bulk transport is not active transport, are you clear?
Yes.
Okay, because active transport is molecular transport, where gradients matter, correct?
Yes.
Here gradients don't matter, right? This is completely different, so learn it properly. Okay, no respectable cell biologist will agree that bulk transport is active transport. Maybe, biochemists who are mostly idiots, they'll say things like this. So, and they call us idiots. This is how it works. And we're all idiots, actually. So, energy is required. Is this making sense?
Yes.
You have to use ATP. Okay. So, bulk transport, BT, is classified into two major modes. Okay. You've got things going into cells, endocytosis, okay, into cells, or exocytosis. What does that mean? Out of cells. Okay. Out of cells has another name. Out of cells has another name, starting with an S. Secretion, thank you. And in fact, secretion is the preferred name, okay? Nobody uses exocytosis. In physiology, people always say secretion, right? So secretion of proteins, particularly, is exocytosis. Secretion of steroids is not. Be careful. Remember, steroids can diffuse through the membrane. They don't need bulk transport, correct?
Yes.
So secretion of proteins. So I'm going to be specific. Oops, writing with the highlighter. Endocytosis is of three modes. You have three modes for endocytosis. Okay. One is phagocytosis. Phago means swallowing. That's why this is called the esophagus. Okay, the swallow bridge.
Yes.
Eso means bridge in Sanskrit. Okay,
Yes.
so the word eso, and they use the same word eso for God. God, bridge, all the same word, right? Because God's a metaphorical bridge. Okay, so phagocytosis, esophagus, right?
Yes.
Bridge, swallow bridge. So phagocytosis is swallowing things. Your immune cells phagocytose bacteria. Okay? So if you don't have enough calcium in the body, you will fall sick very, very often. Are we clear?
Yeah.
Because your immune cells won't function. Because one, they cannot phagocytose things. Two, they cannot secrete things. Calcium is needed for all of it, right?
Yes.
Okay? So please go have a cup of yogurt in calcium's honor tonight. Okay? Have to have your calcium. And for your calcium to be reabsorbed, you need sunlight. Make sense? Vitamin D.
Yes.
So take your calcium seriously. The second mode is penocytosis. Peno means drinking, right? This happens primarily with water. It's the bulk transport of water. What's simple diffusion of water called? Osmosis. This is bulk transport of water. This happens quiet, fast. Are we clear?
Yes.
Cells will not turn this mode on unless they're really dehydrated, okay? Because you're taking water in bulk, right? You don't want to do that under normal conditions, correct? Because it'll dilute all your electrolytes and it's bye-bye time, remember? You don't want that. The last mode is very interesting, and this is how HIV enters our cells. This is called receptor mediated. There's a special passcode. And HIV hijacks part of our membranes, which has that passcode, right? And the first cell thinks that it's yummy cholesterol coming through. Okay?
Yes.
And that's it. It enters and that's it. And now, finally, using CRISPR, people have been able to remove the HIV genome from our chromosomes. Okay, it's still in clinical trials, but it seems pretty promising. Okay. Any questions on bulk transport?
No.
You have to draw this table out, this entire chart of cell transport out, okay, to understand this. This chart won me my very first outstanding teacher award at Berkeley. That was my first award, was for this table, this chart. Every professor uses it there because it's so useful to see all of that information. In fact, they make their students do it during discussion sections. Berkeley has discussion sections, so they make them do this on big sheets of paper. Okay, it's very useful. So draw it, and you'll see all the different components. Okay, the more detailed you make it, the better you'll learn from it. Okay? Any questions on cell transport? Everybody okay?
Yeah.
All right, 5-minute break, 3:45, come back. We will start the next chapter, resting potential. Electricity and potentials. Okay, as scheduled, now we're back on schedule. Okay, yay, all right. So, I'm thinking maybe I should start, I should start another document. Finish it everything. Okay. Okay, so let's start talking about potentials. We're going to be talking a variety of potentials in this chapter, and they're extremely important for the entirety of the course, so make sure you know it. This thing is not on. Okay, is it on now? Yeah. So I kind of have to. No, just easier this way. Okay. So we're talking about potentials. So first let me define what a potential is. Okay? A potential is nothing but voltage difference. Now, if voltage difference is kind of not making sense because it's too abstract, it is abstract. So if you've not had physics, voltage is going to sound weird, okay? I'm going to make it really simple for you, okay? So if you're talking about potentials, okay, again, we're talking about voltage. So voltage equals a difference in potential. So anytime I put a triangle there, that's Greek letter delta, and it stands for difference. Okay? So a delta means difference, so it's shorthand. Okay, so change in potential. What does that even mean? Okay? I'm going to keep it really simple. If you have positive charge, okay, your potential is high. Okay? If you have a negative charge, your potential is low. Okay? Now, if you want to know exactly why this is the case, take physics. Okay? They'll explain it to you, or come see me office hours, I'll show you the derivation. Not that hard, actually. It's just an algebraic derivation, use a little bit of calculus, and I'll show you why that's the case. And I can explain to you why positive attracts negative. There is a mathematical proof for it. Okay, as to why positive attracts negative. Okay, so anyway, that's not the point of this class. So potential is high if you have a positive charge. Potential is low if you have a negative charge, correct?
Yes.
Now, a cell at rest, so cells at rest, have a negative charge inside, okay, and a positive charge outside. Now tell me the potential gradient. Describe the voltage gradient here. So what's a gradient? Difference, right?
Yes.
Whenever I say to describe a gradient, you have to tell me high, low.
High.
Right? So now tell me high and low here.
High from outside the cell, low into the cell.
Correct. So high here and low here. Correct?
Yes.
All right. So this is basically cells at rest. Right? Now, this is a non-zero voltage, meaning cells have a separation of charge. Do you agree?
Yes.
Yes.
And you already saw that, right? The sodium-potassium pump makes sure that sodium is high outside and low inside, and potassium is low outside and high inside, correct?
Yes.
So separation of charge. The scientific word for separation of charge is polarity. Polarity. I'm sure you've seen this in chemistry, right? Polarity. So the cell is said to be polarized. At rest. So the fact that cell is negative inside and positive outside is described as polarized. Are we clear?
Yes.
Okay. That's important because that's how all the other definitions are going to depend on. So anytime I say the word polarized, you should immediately tell me negative inside, positive outside.
Yes.
Yes.
Correct?
Yes.
Careful. Okay, so that's the definition of polarized. Question is first, how is the cell polarized? Well, for one thing, the sodium-potassium pump plays a big role, correct?
Yes.
Because it pumps sodium out, it takes potassium in, and it maintains those, it creates and maintains those gradients constitutively all the time. Now, in addition to the sodium-potassium pump, a cell at rest is slowly leaking potassium out. Okay? Potassium is leaking. So, true or false? Leakage channels are constitutive.
One and a half, yes.
Yes, they're always open, right? That's constitutive. I will use that word on the exam. You need to know how to use that word or you will get that question wrong. Okay? So please make sure you know these words. Okay, so potassium leak channels. Now, understand from chemistry that potassium ions are way bigger than sodium ions, which are way bigger than hydrogen ions. Hydrogen ions are the smallest.
Right.
And I told you, in chemistry, size goes the other way, right?
Yes.
Hydrogen ions are the most powerful. Hydrogen ions can burn your skin. Sodium and potassium can't, okay? If I throw acid on my skin, it's going to burn, okay? So hydrogen ions are the most powerful because of their size, being small.
Yeah.
Then sodium, then potassium. So hydrogen is the smallest, then sodium, then potassium. Are we clear about that?
Yes.
Okay. So if potassium ions are big, okay? Potassium ions are big. This is an important point. Potassium ions are bigger. Potassium is leaking to a positively charged area, correct?
Yes.
Which is the outside of the cell. That's not a favorable thing. Positive doesn't attract positive, correct?
Right.
So that's going to be repelled. So potassium is not welcome on the outside. Makes sense. Important. Which is why it finds the sodium-potassium pump very fast and tries to come back in.
Yeah.
So potassium is leaking to a positively charged area. So this is repulsion. And potassium ions are bigger, right? So you're really big and you move super slow, right? And you're moving to an area you don't want. They all work together. Are you seeing that?
Yes.
Yes.
Always know this. This is a universal truth. Movement of potassium is always slow. What else? Movement was slow. That we saw.
Na plus.
Na plus is never slow. Because Na plus is smaller, right?
Oh.
And it's going to a negative area. So no, Na plus is very fast. What else moves very slowly into cells? Water. Osmosis is slow, right? So remember that. movement of potassium is very slow because of these two reasons. And potassium movement is driven only by a chemical gradient, correct?
Yes.
Guys, and if a movement is driven by an electrical gradient, it can only mean one thing. Positive attracting negative, negative attracting positive. Are we clear?
Yeah.
That's the only time we say electricity drives a movement, right?
Yeah.
Is that attraction force. Here, is this electrical attraction to the outside?
No.
No, it's in fact repulsion, right? So it moves only by a chemical gradient. Contrast this to sodium. Okay? So what happens with sodium? Sodium, first of all, is smaller. It's moving into the cell, correct?
Yeah.
And the inside of the cell is what charge?
Negative.
Highly negative, right? Negative 70, right? Moving into negative charge. Okay? And there was another point and I completely forgot. Oh, the concentration difference is huge. Okay? So sodium, okay, is roughly 145 units on the outside and about 5 units on the inside. Due to what?
Reactive pumps.
Pumps. Sodium, potassium, active transporter, remember? Keeps it low on the inside. So there's a chemical gradient, there's an electrical gradient, and sodium is small. Now tell me, sodium movement is always going to be
Fast.
faster than potassium for sure. Are we clear?
Yes.
That you can guarantee. Okay? So sodium movement is fast. This is going to be very important in action potentials. Very important. Okay?
Yes.
So sodium movement into cells is driven by chemical gradient, yes?
Yes.
Okay, the difference in amounts, yes. Okay, so chemical gradient, obviously, difference in amounts. Also by electrical gradient, correct?
Yeah.
We call this electrochemical gradients, right? It's both, okay? The electrical gradient is the negative charge of the cytoplasm. And its size is small, okay? So all this really facilitates movement of sodium. Why am I not talking about hydrogen ion movement? If you paid attention to acid-base lab last week, you should know this. What do hydrogen ions, what can hydrogen ions usually do? Destroy what? Proteins. Denature proteins, correct? So inside of your cells, full of proteins, you want hydrogen ions sinking in? No. So how does the stomach release the acid? It makes hydrogen ions and instantly transports it out. Are we clear?
Yes.
So when I make these kinds of demands, should I put money down there? If I want this way. Do I have to pay for this? Most likely. So those are ATPase pumps. Make sense? ATPase pumps will grab the hydrogen as soon as it's made, push it out. Otherwise, stomach cells will die. Does that make sense?
Yes.
It's obvious. So keep that in mind. That's why we're not talking about hydrogen ions here, because they're not bioavailable. They're immediately buffered. Make sense? They're immediately grabbed because they're so harmful. Any questions on this? Everyone okay with this so far?
Yes.
Okay. So the next concept is again something that is often very, very confused with students because they don't respect the definition. You paraphrase it whatever way you want. You have no license to paraphrase my definitions. Are we clear? You have to know them word for word as they are. if you want to understand this material, you need to know the word for word. Okay, don't change anything. Try to understand it. Make sense?
Yes.
Okay, so we're going into a concept called equilibrium potentials. So what does the word equilibrium mean to you?
Balance.
Balance, right? Exactly. Equivalence, or things are equal on either side. Yes?
Yes.
Yes.
Obviously here we're talking about concentration. Okay? But let me give you the correct definition, because potential has to do with current, right? So equilibrium potential is the property of individual ions. First of all, it's a property of individual ions. So can I ever ask you, what is the equilibrium potential of a cell?
No.
No, it's rubbish. That doesn't make any sense, right? It's like asking, what's the social security number for this community? Right? So equilibrium potential only applies to specific ions. Everybody okay with that?
Yes.
Yes.
So what is it? Okay, it is the voltage. Please pay attention to how I'm giving you the definition. Intracellularly, meaning, do I care about the outside?
No.
No, I'm only talking about inside the cell. Voltage intracellularly, okay, at which the ion ceases to move across. Now, did I say going into cells, going out of cells? I'm just saying moves, correct?
Yes.
Yes? So for sodium, it's ions moving into cells. For potassium, it would be ions moving out of cells. Make sense? So I'm keeping my definition general. Okay. Now, there's a big formula for equilibrium potential. You don't need to know that. You don't need calculators. You don't need nothing. Okay? You just need common sense. Equilibrium potential is determined by two things: the ion itself and its gradient across the cell. So both of these will determine an equilibrium potential of an ion. Okay, let me explain that. So let's take sodium, for example. So here's your cell. Okay, I told you sodium is high outside and low inside. Correct?
Yes.
Now, the equilibrium potential for sodium is defined as at which point of voltage inside the cell will sodium stop moving. First of all, sodium will move how? If I open the gate, sodium is going to do what?
Enter.
Go into the cell, correct?
Yes.
So sodium influxes. Okay, very important. It goes into the cell. So equilibrium potential, can I say the equilibrium potential for sodium is the voltage at which sodium ceases to enter the cell? Yes, because that's its normal movement, right? So the voltage at which sodium will stop moving or stop entering the cell. You tell me, when will sodium stop? So sodium enters because the inside is super negative, correct?
Yeah.
Common sense. Tell me, when will sodium stop entering?
There.
When it's super positive. Think about it. If the inside of the cell, so the inside of the cell is negative, right?
Yes.
That's attracting the sodium. Everybody got this?
Yes.
Yes.
So if the cell were to be super positive, tell me, would sodium want to enter?
No.
No. Am I making sense?
Yes.
And that number has been measured, and you need to know this number. It is 60 millivolts and plus 60 millivolts. And your textbook is wrong for the 12th edition. I've been making the correction since the 6th edition, telling them to change it. Okay? Because if they're correct, evolution is wrong. Okay? And I know this because the first sentence out of my mouth in my physiology class at Berkeley, the first sentence is, yo, calculate how many sodium ions are needed for action potentials. and everybody sits there and starts calculating. That's the first sentence out of my mouth. Not even good morning. Okay? Because that's the first thing they need to do. It's a 4th-year physiology course. They need to know how to do that. And those people who don't know how to do that will be dropped instantly from my roster. Okay? If you don't know how to do that, that class is gone. Okay? You're not going to pass. So goodbye. Okay? I'll throw them out. So believe me, I know this. 60 millivolts. Are we clear?
Yes.
So at 60 millivolts, sodium will stop entering. Technically 59.8. Close enough. 60 is fine. And you can visualize this, right? If the inside of the cell is 60 millivolts, no way sodium is going to be able to enter. It's going to be repelled. Positive repels positive. Do you agree?
Yes.
Okay. Now, what happens in the case of potassium? So, if you look at potassium, potassium is very high inside, right? And you have a negative charge, right? And potassium is very low outside, and you have a positive charge on the outside. So far, so good, right? This is your polarized state. Polarized means this. Okay. What will make potassium stop moving? First of all, if potassium were to move, which direction will it move? Out. So potassium effluxes. I can't spell. No, that's right. Potassium effluxes. 2 F's. Okay? Do you understand why sodium influxes and why potassium effluxes? E for exit. Good morning. Everybody got that?
Because of the chemical gradient?
Yep. Potassium will leave because of the difference in amounts, right? So, my question is, what will stop the potassium from moving? Now, first of all, what holds potassium there? The negative charge, right?
Yes.
If I make the negative charge super negative, right?
Mm-hmm.
And I'll give you actual values here. So normally it's negative 70, right? This is the rest for most cell types. That varies. For heart muscle, it's negative 90. For your retina, it's negative 30. Okay, so the value varies, right? But typically, on average, it's negative 70, right?
Yes.
At negative 70, potassium leak channels are functioning, potassium will be moving. Are we clear?
Yes.
Now my question to you is, when will potassium stop that movement? Potassium movement is already slow because the proteins and the DNA and the negative charge on the inside and the phosphate are holding the potassium from moving, right? If you're positively charged, why would you want to leave a negative? That's dumb, right? You're attracted to the negative, right?
Yes.
So if I were to increase this to negative 84 millivolts, do you think potassium will say, that's it, I'm not going anywhere? In other words, look, I give you 3 candies, right? And you go, let's make it healthier. I give you 3 broccolis, right? And you go, yum, broccoli, right? But somebody outside the classroom is offering me 10 broccoli florets, okay? Well, in reality, I'm gonna grab your 3 and run out, okay? But the bottom line, potassium can't do that, right? So I'm going to be attracted to that, right? Let's say both these guys are goons, right? They'll beat the crap out of you if you try to take anything from them. But somebody else is offering you more money on the outside, that's where you're gonna go. Are we clear?
Yes.
That's what's happening here. So if the cell voltage becomes sufficiently negative, it prevents all potassium movement. Are we clear?
Yes.
And that number is determined to be negative 84. Negative 84. So at negative 84, I don't care how many leak channels you got. Is potassium moving?
No.
No.
No. So here's a really interesting question. And I'm still not able to find a definitive literature answer to this. Because a lot of literature gives me really funky answers. Heart muscle, potassium is potassium everywhere. Do you agree?
Yes.
Potassium in the heart is not separate from potassium in your tongue, right? It doesn't matter.
Correct.
If the heart and listen to my question very carefully because this could be an exam question. If heart muscle has a resting potential of negative 100 millivolts, it's actually negative 90, but let's say that it has negative 100 millivolts, right?
Mm-hmm.
Tell me, would there be a need for potassium leak channels?
No.
Potassium stops moving at negative 84, right?
Yes.
Right.
If I make these cells negative 100, will potassium even move?
No.
No.
So does it make any sense to have leak channels there?
No.
No.
No, but these leak channels exist. According to literature, first I thought, no, right? I had the same guess last semester. But when I went back and checked in literature, they exist. And now I'm very happy with that answer, that I was wrong. Because that proves evolution. You were wondering why you didn't hear that word for 10 seconds. Evolution makes mistakes. Those potassium channels, according to that literature, they're vestigial. They don't do anything. Okay? Am I
Yes.
making sense? It's of no use, because potassium will never leave anyway. Okay? It's like having that door open and I have a huge sack of broccoli. I'm not moving anywhere. Am I making sense? You don't have to lock the door. I'm not going anywhere. Does that make sense?
Yes.
Yes.
Okay, that type of idea. So keep that in mind. Potassium keeps leaving because the cells are at negative 70. Correct? Here's another test question. Nerve cells, the resting potential is negative 70. Retinal cells, the resting potential is negative 30.
Mm-hmm.
Comment on the potassium movement. First of all, will potassium move in both these cases?
Yes.
Yes, because negative 30 is more positive than negative 70, correct?
Yes.
So potassium will get repelled. Now tell me, will potassium move faster in which condition? In the eye or in the nerves?
The eye.
In the,
In the eyes. Are you seeing that?
Yes.
Because the more positive you are, the more it gets repelled. Somebody's pushing you, right? Assuming the potassium channels are the same diameter. Okay, and as far as I know, they are. Okay? Are we clear about this?
Yes.
yes.
Okay, so this is the kind of thinking stuff I'm going to ask on the exam. Okay? It's not enough for you to just know all this hoopla. You have to know how to take the hoopla and apply it. Make sense?
Yes.
Yes.
Okay. All right. So the equilibrium potential of sodium is plus 60. The equilibrium potential of potassium is minus 84. Note those numbers down. You will need to know it. Our equilibrium potential is okay. Yes?
Wait, so you said potassium stops moving at the number negative 84 millivolts,
Yes.
but you said negative 70 is the rest, so it's
Negative 84 is more negative than negative 70. See, this is, so please, if you have this kind of queries, ask me because it's really important. I promise you, 80% of the class doesn't know this. Okay? Mainly because you need physics to understand this, right? That's why. So remember that negative 84 is more negative than negative 70,
Yes.
and negative 100, which is the heart, is even more negative than negative 84.
Yes.
Okay? So make sure you understand. If you don't, ask me. Those are very important fundamental questions, okay?
Yes.
Always ask. If you're going to be shy and embarrassed and not ask me, you're the one who's going to lose. And I'm guaranteeing you 50% of the class doesn't know this. They're losing also. So you're doing them a favor by asking that question. No question is stupid, other than have you graded my exams? Okay? Or where's my A? I'm going to say turn around. So anyway, now on to the resting potential. But before we go to resting potential, I'm going to give you a quick exercise for you to do. What is the equilibrium potential of calcium ions? Go ahead and do it. I've done it for you, right? Sodium and potassium. I want you to estimate the equilibrium potential for calcium ions. Calcium ions are Ca2 plus,
Yes.
okay? And calcium is 0 inside the cell, okay? It is high. I have a feeling it's 20 outside the cell, but it's 0 inside the cell, okay? That's for sure. And it's high outside the cell, yes?
Yes.
Now do this exact same analysis. You don't have to give me a number. You could just describe it for me. You could also throw out a number for me, okay? So I want you to think, work on it right now for 2 minutes and let me know. What is the equilibrium potential for calcium? Think about what that term means first. If you do it the way I did it, you'll get the answer. Do it step by step. If you do mental math, it's gone, okay? Use your ATPs in your fingers and write all this out. And this unit is most probably the hardest this entire term. So if you think this stuff is hard, you better believe it. Things will get a lot easier from here. Okay. So calcium is high where?
Outside.
Outside, right? So this is calcium. There's zero calcium in the cell, so don't even bother writing it inside. Okay? All right? So if calcium were to go somewhere, which direction will it move? Use the verbiage. Is it going to be an influx or an efflux? Influx, right? So calcium, if anything, is going to be an influx. Do you agree?
Yes.
So calcium comes in because of A, chemical gradient only, B, electrical gradient only, both of them, neither.
C, both.
Both, correct?
Yes.
It's a chemical difference, right? 20 to 0, and electrical, right? It's highly negative inside. Calcium is what charge? 2 plus, right? Divalent, right? That's gonna really wanna come into cells. Do you agree?
Yes.
Okay. So calcium will want to come in. So if you want to block calcium from coming in, what should the inside of the cell be?
Positive.
Super duper positive, because it's a 2 plus ion, correct?
Yes.
Okay, but calcium is a lot larger than sodium. Okay, so there's a complication there, but nonetheless, the equilibrium potential for calcium is 120 millivolts. That happens to be purely coincidental. You cannot argue. Sodium is 60 because it's 1 plus. It's like 1 apple costs 50 cents. What do 2 apples cost? This is not proportions. Are we clear? This is purely coincidental. Okay? It turns out to be 120. Okay? Believe me, if the cell is anything more than 30 millivolts, it will fry. 30 is the maximum voltage that I know of any cell will tolerate. Are we clear?
Yes.
So equilibrium potential of sodium and equilibrium potential of calcium, are they physiological?
No.
No. So would evolution use any of this? No. It's not physiological. But is potassium equilibrium potential achievable? Negative 84.
Yes.
Yes. So of the 3 ions, which one is the most important?
K plus.
30.
Potassium. You can make your calcium concentration tenfold. People live with hypercalcemia, okay, because they have hyperparathyroidism. At one point I had 1,000 times the level of calcium, okay, in my blood. I did, because I had hypervitaminosis D. I have excessive vitamin D, right? It came to control after that. But the point is, body will tolerate any amount of sodium, any amount of calcium. You change potassium even a little bit. I gave my lab class today a fun story, right? The banana frat brother dying story. You change potassium even a little bit, either way, low or high, bye-bye, right? Massive cardiac arrest, okay? Massive. Potassium range has to be controlled very, very carefully, okay? The others, the body forgives you.
You told us that we should have been able to calculate that from what you were going to do. Show us how we should have been able to do that.
Sure, if sodium, it takes 60 millivolts, thank you for that question. If it takes 60 millivolts of energy, electrical energy, to block sodium out, calcium is twice that amount, right? So approximately, it should be around 120. It happens to be 120. Okay, that's just coincidental. Is that making sense? Okay, I know it's 4:20 and I know you're tired. Bear with me. I do feel for you. I'm tired. I've been singing since 7:30. Okay. So I mean, don't feel bad. If you ever want to step out, go wash your face, freshen, or get fresh air, put the recording on and go do it. Okay, don't feel like I'm going to take it personally or whatever. 4:30. Who the hell wants to listen to this? I don't. Okay, so I really sympathize with you. I tried to have this class early, but you can see my schedule. Okay, it's not going to happen. Okay, so is calcium making sense?
Yes.
The equilibrium potential, here's a really interesting one. Let's see if you're able to do this. Okay? Chloride is very high outside. Chloride is very low inside. Rule of thumb, water and chloride are married to sodium. I don't know if I can say that nowadays. Fine, they're lusting after sodium, always. I can say that. That's politically acceptable. Don't get married. Okay, so old school. Okay, so chloride follows sodium, water follows sodium. You need to know this. Okay? So if sodium is high outside the cell, guess who's going to be high outside the cell? Chloride, correct? Yes?
Yes.
What is the equilibrium potential of chloride?
60?
If you're answering that quickly, you're not thinking at all. This is a negatively charged ion, correct?
Yes.
Would a positive charge inside stop a negative charge from entering?
No.
Go back to the definition. Do not pull this on the exam. Think, then answer. Process, then answer. That's what I'm trying to teach you. Because guess what 102 did? They made you like Jeopardy, right? Sooner you answer, smarter you are. Nope. Think, respect the question. That's not an easy question. You want, first of all, you want to think, first of all, the cell is normally what?
Negative.
Negative inside and positive outside, right?
Mm-hmm.
Now, you cannot argue if you make the outside positive enough, chloride would not want to move, right?
Yeah.
You cannot do that because what's the definition of equilibrium potential? Look at your notes.
Intracellular.
Intracellular, bingo. You're looking, now do you see why I put that word there? Okay? So, outside voltage, who cares, right? Tell me, when will chloride stop coming in? It's driven by concentration gradient, right? Is it driven by an electrical gradient? Will chloride be attracted to negative?
No.
No, it's not. Same problem as potassium, right? It is not driven by an electrical gradient. It's only driven by a chemical gradient. So tell me, will it be fast or slow?
Slow.
Slow. There's only one thing pushing it, correct? So if chloride were to be stopped, so question is, why is chloride even moving? Chloride's moving because the inside is negative, right?
Yes.
Not negative enough, correct?
Yeah.
If I made the inside super negative, chloride would get repelled. Do you agree?
Yes.
Yes.
Yeah. So that's what happens, except it turns out the equilibrium potential for chloride is the exact same value as your resting potential. The equilibrium potential for chloride is negative 70. That's when chloride stops moving. Okay, this is experimentally determined, right? You would have expected it to be negative 80, right? Or negative 85, or negative 90. Turns out it's negative 70. So tell me, at rest, does chloride move?
No.
No. Very, very important. At rest, chloride does not leak into the cell. So is there any point to having chloride leak channels?
No.
No. So chloride leak channels do not exist. Are we clear?
Yes.
Because it's physiologically unnecessary. So if I put that as an answer choice, I don't care what the question is, unless the question says which of these is a cell lacking, right? You will ignore it, correct? Chloride leak channels don't exist. Is this making sense? You need to memorize all these values. You need to know these values. Yes?
Yes.
The last concept I'll leave you with before we start back on Thursday is voltage changes. Okay? And before I go to that, so bear with me, I want you to stay till I finish. It will only be 2 minutes, okay? Yeah, that's what he always says. I can hear your mental voice. Okay, so the resting potential for most cells, okay, vary. Okay, so the average is -70, and I need you to know that. Millivolts. Okay, number one. Number two, okay, so at -70 millivolts, okay, the cell is polarized. Okay, polarized means this, right?
Yeah.
Okay, now, when sodium influx happens, tell me, sodium influx will make the inside of the cell less negative, more negative?
Less negative.
Less negative, right? Will make cytoplasm less negative. Now, being negative is polarized, right? So the opposite would be depolarized. Activating, deactivating, right?
Yes.
The minute you put DE, that means it's an opposite in Latin. You're making it the opposite, depolarizing. Then you hit a maximum, and I already told you what that value is. For all cells, maximum is
Plus 30.
plus 30, thank you. You hit maximum, and this is called amplitude. Okay? Now, the cell then starts leaking potassium out. Okay? And we don't say leaking, we say potassium efflux. Okay? Will decrease. So at amplitude, you've got this, right? The cell is positive inside, negative outside. Are we clear?
Yes.
That's why it's plus 30. So will decrease the cell voltage. Okay? And you go back to this, correct? You go back. I am looking back at my notes. I am reviewing my notes. Reviewing. Looking at it again. Reviewing. So the term would be repolarize. But potassium doesn't stop at -70. What does it go until? -84. Because that's its equilibrium potential. So you make it excessively negative. What's the word for excessive? I was screaming this last week. Hyper, right? So what would you call this? So excess negative inside would be hyperpolarization. And then you return back to rest. Everything closes, and sodium potassium pump will return everything back to rest. Fast or slow?
Slow.
Very slow. Am I making sense? Very slow. So that process is called restoration. And with that, class dismissed. There are all the terms. And we're going to start with action potentials Thursday. Okay?
Yeah.
Goodbye.